All right, I want to go ahead and get started. Hello, good afternoon. I am Luis. You guys all know me. This is our Unit 1 celebration of knowledge review. This is just an extra review to kind of help you prepare for Unit 1. And I'm going to cover items that typically tend to be missed or forgotten by students on the Unit 1 exam. And so I'll just kind of do a brief overview, go over some of the key highlights, give you some tips, and then be open up for any questions. There we go. Okay, so for the exam in general, it's going to be worth 80 points. You're going to have about 90 minutes to complete it. It's going to be about 18 to 20 questions, depending on how many questions Dr. Cotter puts in there. It's going to be a combination of multiple choice and short answer questions. The exam is going to open on Thursday morning, and it'll close Saturday night at 11:30 p.m. And so what that means, if you start the exam a little late, at 11:31 p.m., it's going to kick you out, and whatever you complete is what gets submitted, and whatever you don't complete, you get a 0 on. So the latest you should be starting this test is 10 p.m. I would honestly try to do it sooner. That way you give yourself time in case you have any technical errors, and so you don't miss time on the exam. The exam does require a Respondus lockdown browser. There is a quick 2-point assignment to test Respondus in the lecture Canvas course, so go ahead and do that. If it's not working on your laptop or there's certain requirements, like you don't have a camera on your computer, you can come and take the test here on the school computer in the library. They are open Saturday, but they're only open from, I believe, 10 to 2 p.m. You'll have to check those times, but it's a very short, brief window on Saturdays. Or you have to make other arrangements like borrowing a laptop from someone else or going to another public library. If you're going to be using an iPad, I created a YouTube video for how to log into the test using the Respondus app, and then you can sign in to your Maricopa Canvas and then get to the exam that way. And as always, if you have any technical issues, please let Dr. Cotter and I know as soon as possible. And then lastly, make sure you take this test in a quiet place, preferably not on a table, preferably on a table and something solid. Don't do it on your lap. Don't do it in the kitchen with a bunch of people walking around. Give yourself the best opportunity to succeed on this test. And to do that, you want to be in a quiet environment, someplace that has good internet connectivity, and just in general, a place you won't get distracted, like your Starbucks or at home. You know, you have to put the kids down first and then go take the test, do that. But try to give yourself a good optimal condition so you can focus on the test. Okay, so I'll freeze this. Okay, so first thing I'm gonna quickly go over is Koch's postulates. So this is 4 weeks ago now. A lot of times we forget Koch's postulates, kind of forget that we went over this, and sometimes it gets missed when you're reviewing for the exam. So very quickly, the 4 steps: make sure you observe the patient for signs of illness, and you isolate the microbe from the affected host. Just remember that signs are what can be seen, such as temperature readings, fevers, any kind of wounds, and then symptoms are things the patient feels, like nausea, upset stomach, that kind of thing. So if the question asks you for Koch's postulates, to state the first step, and if it asks you list 2 signs and 2 symptoms, you should be able to distinguish between signs and symptoms. Step 2, culture and identify the microbe in the lab using different tests. For this one, I always tell you to pick 2 or 3 that you like and that you can remember, and those should be your go-to tests for any of these questions. So I like macroscopic, very easy to describe. Microscopic is looking at cell morphology under a microscope. Macroscopic is looking at colony morphology. So be able to pick 2 or 3 that you really understand and just be able to quickly describe that specific lab test. Step 3, infect a new host with the same microbe and observe the same signs of illness. That's pretty self-explanatory. And then step 4 is one that people tend to miss for some reason. You gotta remember to perform the same exact lab test. So don't get cute and do 2 for step 2 and then 2 different ones for step 4. Whatever you pick in step 2 should be identical to step 4. So just don't forget those points. Okay, so we kind of started going over this yesterday, but cell parts are a big part of the study guide. If you go look at things like in pages 2, pages 3, pages 6 and 7, cell parts are probably going to be asked, and not in the way you think. So the question might not say, hey, what's a flagella? What you're more likely going to see is a question that might ask you, you get lab results back, and under a microscope, you see a microbe that has a cell wall and a cell membrane. What could your potential suspects be? And in this case, you can see I use this one as a comparison for fungal cells versus bacterial cells. You can see they both have cell walls and cell membranes. So in a question like this, you would say it could be a bacteria or a fungal cell based on the information that's given. But here's just a quick look around of what some different cell parts that we have in bacteria, fungal cells. Also remember, see if I can use it. But another thing I like to focus on is like naked versus enveloped viruses, right? You see the similarities, you see a capsid, you see the DNA or RNA, but never both. And you also see the viral spikes. The envelope virus has the same thing, a capsid coat, DNA or RNA, which is labeled genome here, and the viral spikes, but then you also have the viral envelope on the outside. So again, the questions are not going to be, hey, what is a viral envelope? What is a viral spike? It's going to be more so like your case studies. Hey, you see these specific features. What could your possible suspects be? So we did go over this yesterday. I would definitely just make sure that you're able to identify these different cell parts and then associate them with specific things like the bacterial cells, fungal cells, and viruses. Again, I talk about cell part functions. Just be able to, look at some of understand what the cell parts are for. You might be given questions like, hey, give me 3 cell parts of a bacteria and their functions. And so a question like that, you have the freedom to choose any 3 that you want to choose. So just make sure that you study the virus part, or the cell parts and their functions. That way you're able to answer a question that might ask you to pick 3 and also describe their function. Okay, bacterial cell shapes and flagella types. Again, a question like this might be more of a picture. So you might be given an image of any of these bacterial shapes that we discussed. I would usually focus on the Staphylococcus, Streptococcus, Streptobacillus, the Spirillum, the big 3 that we really talked about in lecture. But if given an image, you're going to be asked to identify a certain bacteria. And the question might say, hey, you see this under a microscope, and say it gives you this Streptococcus. It's going to ask you what kind of bacteria is this? And you'll have the option Streptococcus bacillus, Streptobacillus, Streptococcus, Staphylococcus, Spirillum, etc. So just be able to associate the images with that specific bacterial type. And the same thing for the flagella. You might be given an image on one of these, and you're going to be asked, okay, what type of flagella type is this microbe? So just go ahead and study this so that you can answer it properly. A big one as well, kind of going back to the part where this exam is very case study-like, and so where the questions are going to ask you, hey, you get these results from the lab, and you're going to see, they're going to tell you that this microbe stains purple, and it has teichoic acid. What kind of microbe could this be? And so being able to distinguish between gram-negative and gram-positive bacteria, mold versus yeast. Give me 1 second. Mold versus yeast, bacteria versus fungus versus protozoan, and then the types of viruses, naked versus enveloped versus complex or bacteriophages. if the question might ask you to, hey, give me 3 things from a gram-negative and 3 things from a gram-positive bacteria. It could give you the parts or the differences things associated with a yeast cell. So you got to be able to identify a yeast versus a mold. Again, most of Unit 1 is being able to be given parameters of a cell or a type of microbe, like the cell wall structure, color stains, that kind of thing, and be able to pick out whether it's a mold or a yeast. On this one specifically, does anyone in the group have any questions? And if not, the way you can find those for all of these, that's going to be page 2, pages 3, page 5, page 6, and page 7 of the study guide. So you can kind of see it's about half our study guide is just being able to compare and contrast all these different microbes. So you should be very comfortable being able to do that. Okay, the last one I'm going to go over, this one's also a very heavily missed question, especially on chapter 6, is viral infection and replication. So obviously the first one is to remember your steps: attachment, entry, encoding, replication, assembly, release. But you got to be able to talk about the specific differences between the 2 viruses. So in the case, I'll use HIV. You've got the GP120 and GP41 viral spikes. They infect CD4 plus helper T cells, and they utilize reverse transcription or reverse transcriptase, which is the enzyme. And then for influenza, you've got hemagglutinin and neuraminidase viral spikes. They infect respiratory epithelial cells, and they use RNA polymerase. You can see here on the right, I chose the replication cycle of HIV. And even if I just got this image with no other information, I could tell it's HIV because I was given that it's a CD4 cell and that it undergoes reverse transcription. But be very comfortable being able to describe viral replication in detail for either HIV or influenza. You might be, I guarantee it's probably going to be on the exam. I just don't know which one of these it's going to be. So you've got to be able to be comfortable doing it. Dr. Cotter's going through the chapter 6 case studies. So if you missed points on this question specifically, I'll go back and figure out what you missed so that you can get it right for the exam. Those are really the big highlights I wanted to go over. Again, most of it for the exam, anything on the case study is fair game. But the things I see people miss the most are always the viral replication, Koch's postulates, and just in general, the cell parts. Some of the other things you should be able to get, function of viral spikes, they recognize, they attach, and they allow for entry. Those are pretty self-explanatory. Endospores, be able to discuss what an endospore is, the 3 layers, what they're made of, and why certain bacteria have endospores, what conditions allow them to become endospores. And that's pretty much it. It's not too difficult. I think Unit 1 is one of the easier ones to go through. As we start going through the rest of the semester, you'll get into like chemical, physical methods to kill microbes, and then you're going to talk about the immune system and the different types of things our immune systems can do to protect us. So Unit 1's a pretty good one to, pretty easy one to kind of study, look over, and then that way you get a good grade now that can help you take any bumps you might get from Unit 2, Unit 3 exams. Besides that, does anyone have any questions that I didn't go over? Or have anything in general about the exam, or just what to do, when it's due, that kind of thing? And then for anyone that just joined, I did see some people just joined halfway through. Biggest thing I want to highlight is this information right here. I did send an announcement on it as well, so you have the information. But the exam is 80 points. You have 90 minutes to complete it. It's going to be multiple choice and short answer. And you're going to have from Thursday morning till Saturday night at 11:30 p.m. The exam will close at 11:31. So if you are in the middle of the exam, it will save what you have and then submit it. So try to get that started earlier. It does require Respondus, and we have a Respondus quick 2-point assignment so you can test Respondus on your computer. But if you know it's not going to work for you, if you have an older version of an older computer, older software type, I would definitely try to get a laptop or a stool computer so you can take the exam. And again, just pick a good location. I'm in the library right now, rented out a room, or another quiet place in your home that allows you to take the test uninterrupted. That way you can focus on the test, not worry about dogs barking or kids or whatnot. Besides that, I'm just going to leave you with this. We do have President's Day on Monday, so Monday we do not have class. But a good quote from one of our old presidents: "I find that the harder I work, the more luck I seem to have." You're not going to luck into a good grade. All those grades come from your work and how much effort and attention you put into studying. So what I would look at is, if you feel unconfident with anything, go back and look at that. If there's a topic that you think 100%, don't study that now. Go and study what you're not sure about, and then go back and refresh on the stuff you do know. That way, when the exam starts, you have a very good grasp of the material, and you can just kind of go through it, power through it. I think on average, I've seen some people take this test in about 20 to 25 minutes for people that really understand it. And most people take about 60 minutes, but you are given 90 minutes if you want to take the extra time. Other things that you can look at is if you get to a question and you're not really sure what to do with it, just skip it, go to the next one. Hopefully something later on in the test will help you remember what that question could have been. But worst case, sometimes it's easier to do the questions you get a little bit of a positive reinforcement that you can do it, and then you can go back and answer those tough questions. One of the questions I have in the chat: will the test include anything from page 1 of the study guide? That is a good question, Krista. And yes, so I mentioned Koch's postulates. So you should be able to state the 4 Koch's postulates. And the big things that I want to highlight is you should be able to distinguish between what is given to you, what is a sign, and what is a symptom. And then in steps 2 and steps 4, you should be able to pick 2 or 3 of these tests. We're not going to select them for you. Usually you can pick the 2 or 3 that you want to use, but they should be exactly the same in steps 2 and steps 4. So people miss points because they may have the right test in step 4, but it's different from step 2 and you'll lose points. Okay, awesome. Glad I could answer your question. That's all I have. Does anyone else have any questions, any last minute comments or concerns? Okay, if not, I'm going to stop recording and let me go back here. And I'm going to wish you guys all the best of luck. Like I said, you don't really luck into the test. You have to study. But I'm available for any last minute questions if you want me to review anything for you before we open up for the exam. Thank you and good luck.